Hi friends, my name is Akhilanand Singh. Welcome to another video on biological anthropology. It covers Unit 9.4 of the UPSC Anthropology Optional Paper 1. This video is all about chromosomal aberrations. It has 4 sections. The first section discusses various aberration types. The second section talks about syndromes arising out of aberrations. The third section focuses on genomic imprinting, and the final section discusses avoidance techniques for chromosomal aberrations. Chromosomal aberrations are changes in the structure of chromosomes or number of chromosomes. Chromosomal anomalies occur due to errors in cell division following mitosis or meiosis. Spontaneous mistakes during cell division, environmental mutagens, and late pregnancy are common causes of chromosomal aberrations. Karyotype testing detects chromosomal abnormalities. Most fetuses with chromosomal abnormality do not survive beyond the first trimester of pregnancy. Parts of a chromosome. Let's revise our fundamental knowledge of chromosomes and their structure. Chromosomes reside in our cells. Humans have 23 pair of chromosomes. We get 23 chromosomes from each of our parents. 2 chromatids make a chromosome. Chromosomes have short and long arms denoted by the letters P and Q. The place where chromatids join is called centromere. The outer ends of the arms are called telomeres. Each arm of a chromosome has regions, bands, and subbands in nested fashion. The regions, bands, and subbands are numbered from centromere to telomere in increasing numbers starting with 1. All of our chromosomes are not the same in shape and size. They are structurally different and also vary in length. Based on the point where chromatids join, the human chromosomes are called metacentric, submetacentric, and acrocentric. Metacentric chromosomes have the centromere near the center. Submetacentric chromosomes have the centromere slightly away from the center. A chromosome where the centromere is near the end of it is called a acrocentric chromosome. Humans usually have 5 pairs of acrocentric chromosomes numbered 13, 14, 15, 21, and 22. The Y chromosome is also acrocentric. Metacentric chromosomes include chromosomes 1, 3, 16, 19, and 20. Chromosome 1 is the longest human chromosome. It is 3 times longer than the chromosome 22. Chromosomal aberrations are of several types. Numerical and structural aberrations are their broad level classification. Numerical chromosomal aberrations occur when whole chromosomes are lost or extra chromosomes get added to the standard set of chromosomes. This results in a change in the total number of chromosomes from 46 to something else. Structural chromosomal aberrations arise when one or more individual nucleotides are lost or extra nucleotides get added to a chromosome. It makes the individual chromosomes shorter or longer than their typical size. In structural aberrations, the number of chromosomes does not change, it stays at 46. Numerical aberration types. Euploidy and Aneuploidy are further divisions of numerical aberrations. Monoploidy, diploidy, and polyploidy are subdivisions of euploidy. Hypoploidy and hyperploidy are subdivisions of aneuploidy. Let us understand all these terms one by one. Numerical aberration, euploidy. In euploidy numerical aberration, human cells have chromosomes in the multiple of 23. That is, they have 23 chromosomes, 69 chromosomes, 92 chromosomes, and so on. Having 46 chromosomes in a cell is normal, it is not an aberration. Let us understand the term ploidy first. Ploidy means the number of complete set of chromosomes in a cell in an organism. Normal human cells are diploid. It means it has 2 sets of chromosomes. A cell with any number of complete chromosome sets is called a euploid cell. Euploid can be divided further into monoploid, diploid, and polyploid. A human monoploid cell has a single set of chromosomes. It means it has only 23 chromosomes. Diploidy is a state in which cells contain 2 sets of chromosomes. Normal human cells are diploid. It means a diploid cell has 46 chromosomes. Polyploidy is the state where cells have multiple sets of chromosomes beyond the basic set, usually 3 or more. Triploid is a specific case of polyploid when cells have 3 sets of chromosomes. Other cases are tetraploid with 4 and pentaploid with 5 sets of chromosomes. Numerical aberration, aneuploidy. Aneuploidy is the presence of an abnormal number of chromosomes in a cell. For example, a human cell having 45 or 47 chromosomes instead of the usual 46. It does not include a difference of 1 or more complete sets of chromosomes. A cell with any number of whole sets of chromosomes is called a euploid cell. About 68% of human solid tumors are aneuploid. Most cases of aneuploidy in autosomes result in miscarriage. Autosomal aneuploidy is more dangerous than the sex chromosome aneuploidy. Numerical aberration, aneuploidy. Aneuploidy can be divided further into hypoploidy and hyperploidy. Hypoploid cells contain 1 or more than 1 fewer chromosomes than the standard. A loss of a single chromosome is called monosomy, and the loss of both pairs of homologous chromosomes is called nullisomy. Hyperploidy is a state in which cells contain one or more extra chromosomes. The gain of an extra copy of chromosome is called trisomy, and the addition of two chromosomes is called tetrasomy. Structural aberration types. Let us now talk about structural aberrations. In structural aberrations, the length of some individual chromosomes change. The total number of chromosomes stays at 46. Insertion, inversion, duplication, deletion, and translocation aberrations are further division of structural chromosomal aberration. In insertion chromosomal aberration, one or more nucleotide base pairs are added to a chromosome. In inversion aberration, a piece of chromosome detaches and then attaches to the same spot but in the opposite direction than it was initially. When there is an increase in the number of copies of a gene within a chromosome, the aberration is called duplication aberration. A segment of chromosome goes missing in deletion chromosomal aberration. The translocation aberration occurs when a chromosome breaks into fragments and fragments fuse onto a different chromosome. Examples of chromosomal aberrations. This section of the video discusses some common chromosomal aberrations in humans. Some examples are related to sex chromosomes and some to the autosomal chromosomes. Just to recap, sex chromosomes are X and Y chromosomes and the rest 22 pairs of chromosomes are autosomal chromosomes. Sex chromosomal aberration, Klinefelter syndrome. Klinefelter syndrome is a type of hyper-aneuploid XXY trisomy aberration. Persons having this syndrome have two or more X chromosomes with one or more Y chromosomes. It impacts males who have small testes, tall stature, lack of secondary male characteristics, gynecomastia, and male infertility due to reduced sperm count. They often have mild mental retardation. In lab tests, such males have shown low testosterone levels. Sex chromosomal aberration, Turner syndrome. Turner syndrome is a condition that affects only females. It arises when one of the X chromosomes is missing or partially missing. It belongs to the numeric chromosomal aberration of hyperploid aneuploid type with X monosomy. Turner syndrome can cause a variety of medical and developmental problems, including short height, failure of the ovaries to develop, infantile genitalia and breasts, and congenital heart defects. Lab findings report decreased estrogen production. Sex chromosomal aberration, Superfemale XXX syndrome. Superfemale syndrome or the XXX syndrome is a chromosomal disorder that exclusively affects females. Instead of the standard XX chromosomal pattern, the individuals with triple X syndrome have an additional X chromosome. It belongs to the numerical chromosomal aberration of the hyper-aneuploid type with XXX trisomy. It is characterized by delayed speech and language development, learning and cognitive challenges, and menstrual irregularity. It also brings in behavioral and emotional difficulties along with physical abnormalities such as tall stature, flat feet, and mild motor coordination difficulties. Sex chromosomal aberration, Instersex Disorder. Many chromosome configurations other than simple 46XX or 46XY can result in disorder of sex development. Intersex is an umbrella term used to categorize various reproductive and sexual anatomies that are ambiguous and don't fit the typical binary notions of male or female bodies. Intersex individuals may have chromosomes, genitals, or internal reproductive organs atypical of male or female definition. Sometimes they may possess characteristics of both male and female sexes. Intersex by definition is when someone appears to be one sex but has dominant anatomy of the other sex, or when someone is born in between the typical male and female sexes. Autosomal chromosomal aberrations, Down syndrome. Down syndrome is named after John Langdon Down, the British physician who described the syndrome for the first time in 1866. The primary cause of Down syndrome is chromosome 21 trisomy, a numeric aberration of the hyper-aneuploid type. Individuals with Down syndrome have severe mental retardation, short hands with a single crease on the palm, a flat face, wide set eyes, white spots on the periphery of the iris, and congenital heart defects. Autosomal chromosomal aberration, Patau Syndrome. Individuals with Patau Syndrome have severe mental retardation, small head, cleft lip and palate, small eyes, congenital heart and renal defects, umbilical hernia, and rocker bottom feet. Most cases of Patau Syndrome are caused by trisomy 13. Dr. Klaus Patau and Dr. Eva Thurman ascertained the chromosomal nature of the Patau Syndrome in 1960. The disease is named so in Patau's honor. Autosomal chromosomal aberration, Edwards Syndrome. Most cases of Edwards syndrome are caused by trisomy 18. The likelihood that parents will have a child with Edwards syndrome increases with maternal age at the time of pregnancy. Severe mental retardation, rocker bottom feet, small jaw, low set ears, congenital heart and renal defects, and overlapping fingers are the defining physical characteristics of this syndrome. Only 5 to 10% of children survive beyond their first year. The disease is named after the English geneticist John Hilton Edwards, who described the syndrome in 1960. Autosomal chromosomal aberrations, Cre-Du-Chat syndrome. Cre-Du-Chat syndrome, also known as 5p- syndrome, is a chromosomal condition that results when the short arm of the chromosome 5 goes missing. It is a structural chromosomal aberration of deletion type. Cre-Du-Chat syndrome patients have severe developmental delay, cognitive defects, and distinctive facial abnormalities like round face, low set ears, flattened nose, eyes spaced wide apart, and a small head. Infants with this condition often have high-pitched cry that sounds like that of a cat due to structural abnormalities in the larynx. The French doctor Lejeune described the disorder first in 1963. Cre-Du-Chat translates to cry of the cat in French. Genetic imprints in human diseases. Let's now talk about genomic imprinting. UPSC question papers often include a question from this topic. For every gene we get two alleles, one from the mother and the other from father. Having two alleles come in handy in case when one allele gets damaged, the other takes over. Genomic imprinting is a process of silencing or inactivating genes. DNA methylation and histone acetylation are two processes responsible for it. When imprinting happens, the imprinted allele becomes inactivated or switched off while the other allele stays active. It leads to a situation where despite two alleles of the same gene being present, they behave as if there is only one allele. The silenced one is as good as non-existent. Left with no substitute allele, the single active allele is highly vulnerable to mutations. Chromosome 15 is especially prone to genomic imprinting. The gene imprinting happens in the region Q11 to Q13 of chromosome 15 during the development of germ cells into sperm or egg cells. Example of genomic imprinting disorder, Prader-Willi Syndrome. Prader-Willi Syndrome occurs when the alleles of a specific gene from the mother are imprinted or silenced and the alleles of the same gene from the father get deleted. It happens in the Q11, Q13 region of chromosome 15. With one allele silenced and the other deleted, the zygote has no genes left to make the essential protein. This situation brings in a significant clinical condition. It results in mental retardation, impaired language skills, early childhood obesity, feeding difficulties during infancy, characteristic face like small upward nose, thin upper lip, short stature, small hands and feet, and growth hormone deficiency. Genomic imprinting disorder, Angelman Syndrome. Angelman syndrome occurs when the alleles of the gene UBE3A on chromosome 15 from the father are imprinted or silenced and the corresponding alleles from the mother get deleted. With one allele silenced and the other deleted, this zygote also has no genes to make the essential protein, and the situation causes a significant clinical condition. It results in mental retardation, puppet-like demeanor, like frequent laughter and smiling, often with little stimulus, being easily excitable, frequent flapping of hands, impaired language skills, seizures, and neural development issues. Prader-Willi Syndrome and Angelman Syndrome compared. Note the difference in the causes of these two syndromes. In Prader-Willi Syndrome, the allele from the mother gets imprinted and father's alleles get deleted. In Angelman Syndrome, the alleles from the father gets imprinted and the mother's alleles get deleted. Genomic imprinting, anthropological view. The number of human diseases due to genomic imprinting is more than 100. It includes diseases like cancer, obesity, and diabetes. Prehistoric evidence suggests that genomic imprinting evolved about 150 million years ago. Recent years have seen an increase in the incidence of various imprinting disorders, especially among children conceived using assisted reproductive technologies. Chromosomal aberrations, avoidance techniques. We discussed various types of aberrations. Now let us talk about ways to avoid them. Genetic screening. Genetic screening is a term used to help us identify a smaller group of people from a large population. This smaller group of people might have a higher risk of either having a disease, developing that disease, or potentially having children who may have that disease. Genetic screening is different from genetic testing. Genetic testing focuses on an individual and genetic screening focuses on whole population. Genetic counseling. Genetic counseling refers to guidance related to genetic disorders that a specialized healthcare professional provides to an individual or a family. A counselor might interpret genetic tests designed to help estimate the risk of a disease. The counselor addresses the concerns of an individual or family and helps them make informed decisions about their medical condition. They provide psychological counseling to help them adapt to their condition or risk. Human DNA profiling. DNA profiling or DNA fingerprinting is a technique of uniquely identifying people based on their DNA characteristics. Although 99.9% of human DNA sequences are same in every person, enough DNA is different. This different part of the DNA allows us to distinguish one individual from another unless they are monozygotic or identical twins. Uses of DNA profiling. Criminal investigators use DNA profiling as a forensic investigation technique. They compare a suspect's profile with the available DNA evidence to assess the likelihood of their involvement in the crime. It is also used in paternity testing, to establish immigration eligibility, and in genealogical and medical research. Gene mapping. Gene mapping is the process of determining the location of genes on chromosomes. A gene's specific location on a chromosome is known as the locus. The locus can be used as a molecular marker to find the distance between the other genes on the same chromosome. Gene maps allow researchers to predict the inheritance patterns of specific traits, which can eventually lead to better understanding of disease-linked traits. Today, the most efficient approach for gene mapping involves sequencing a genome and then using a computer program to analyze the sequence to identify the location of genes. A gene map of all the genes of humans from both physical and functional standpoint is called the Human Genome Map. Time to end the video. Let me know if it is helpful for you. Thank you.